we're going to get back going, keep it rolling. Next up, please welcome Daphne Banerjee to the stage. Daphne is the founder and CEO of ImmuNovX Biotech. Daphne.
Have my trusty glasses here in case I actually have to see anything. Okay, there are the slides. Perfect. Hi, I'm Daphne Banerjee. I'm the founder and CEO at ImmuNovX Biotech. And I'm thrilled to be here today and looking forward to this discussion. Oh. Okay. Can you guys see the slides? Okay, perfect. All right, so ImmuNovX is focused on antigens. Antigens, as you may know, are the engines that make vaccines work. You can think of an antigen as the active ingredient in a vaccine. The better the active ingredient is, the better the vaccine. And so antigens drive vaccines. Which way do I point this? Okay. But there are issues with antigen production. They're time-consuming and costly to make. In fact, this is important because antigen production is about 50% of the entire cost of manufacturing a vaccine. They're complicated to make, and there are suboptimal efficacy and safety issues, and there are cold chain issues. If utilization of some antigens, some types of antigens, will make vaccines that require extremely cold transport and storage. Where do I point this? Okay, and that's where ImmuNovX comes in. We've solved the problem of antigen production. Our process is quick. It takes only 2 hours versus weeks or months, while lowering costs by about 25%. Vaccines that use our antigens are simple to make, one step versus multiple passages. Our antigens have two very important characteristics. They're both live attenuated, which makes them highly effective, and replication defective, which makes them safe. In addition to that, vaccines that use our antigens require fewer cold chain constraints, and this is a major issue because cold chain can add 34% to the cost of manufacturing a vaccine. So you might be asking yourselves, who cares? Who cares about all this? Well, vaccine manufacturers care, and they are our target customers. Vaccine manufacturers will want to license our technology because because of all the things I just said, but also because it will allow them to make better antigens more quickly and save them money. So I mentioned and will save them money. There are other beneficiaries of our technology as well, as demonstrated here. I still haven't figured this out. I'm so sorry. There it goes. So let's talk a little bit about the science. This is how our platform technology works. You have a pathogen that contains DNA. You treat that pathogen with our secret sauce, centanamycin, which alkylates or binds up the DNA. Once you wash away the centanamycin that's not bound to DNA, you're left with an almost dead antigen that's also unable to copy itself. This antigen is now capable of eliciting a powerful immune response. We know this works because we have POC in vitro and in vivo data across pathogen types and animal models. We also have challenged immunized mice. We've demonstrated both virus neutralization and dose dependency. Case in point, here we have two graphs, herpes simplex virus 2 on the left and cytomegalovirus on the right. Both pathogens have been treated with varying concentrations of centanamycin, and both pathogens demonstrate dose dependency, or both experiments do. Here we can see that mice were mice were protected against live virus challenges. We have a One Health approach. If you're not familiar with that, One Health just is that animal health, human health, and the environment are all intertwined. What this means for us is that our growth strategy is basically going into the veterinary market first and then into the human market. There are three other really good reasons to go into the veterinary market first. One is the process is a lot simpler, as you probably know. Because of its relative simplicity, we will begin to see revenue in about 2.5 years, which we could never do going through FDA, but we will get there eventually. And finally, the early studies that we do will be used or leveraged for human product development as well. We've spoken with or interviewed nearly 150 experts about this. They have confirmed our approach, and they've confirmed our approach and validated the fact that there's a clear regulatory pathway for both veterinary and human development. Oh. The veterinary vaccine market, the global veterinary vaccine market, is predicted to be worth $15 billion by 2027, and we aim to capture a sizable $274 million part of it with licenses within 5 years of launch. The total global human vaccine market is predicted to be worth $92 billion by 2027, and our goal is to capture almost $700 million chunk of it, almost $700 million chunk of it, with licenses, again, within 5 years of launch. There it goes. Okay, so our main competitor is a startup company. It's called Anaviv. They have a One Health approach as well, and they are focused on coccidioidomycosis. You'll hear me say that a lot, but that's our first pathogen as well. In addition to Anaviv, there are three companies we consider competitors. They either have a platform technology that engineers antigens like we do, or they have a licensing model or expect to use a licensing model like us. The licensing model actually has been tried and true over the years. In fact, several companies in this space have been acquired by the likes of J&J, Novartis, and Baxter. We're seeking $875,000 by way of a SAFE. We're also open to a convertible note if the lead investor would prefer. We're seeking lab space and partnerships as well. So we have so far garnered $156,000 in non-dilutive funding, including my contributions. We recently submitted grants to NIH and BARDA that total $463,000. As I mentioned, we're currently raising $875K, and those funds will be used to support our synergistic studies, and I'll tell you more about those later, but the synergistic studies will help us get to what I call a multi-pronged inflection point. So after the synergistic studies have been completed, development will go rather quickly, or veterinary development will go rather quickly in coccidioidomycosis. Now, you might not have heard of coccidioidomycosis, but it is a very important disease. It's on the NIH's radar for a vaccine, and it's rare. So there's the potential for an orphan drug designation. In fact, the California Department of Public Health is right now investigating an outbreak of coccidioidomycosis in folks who attended a recent music concert. So then we'll need about $7 million to complete veterinary work and begin selling licenses to vaccine manufacturers in late 2026. We will take our technology to phase 1 and look for a complete or partial buyout or a strategic merger before 2030, at which time we will exit. This is just a breakdown of the use of funds, or the way we'll use the funds to get to that multi-pronged inflection point I've been talking about. And the way we get there is by synthesizing, doing 5 things, is synthesizing attenuated coccidioides strains, by graduating centanamycin's attenuating effect, demonstrating antigen immunogenicity. We'll also identify a surrogate biomarker, and this is important. We will also demonstrate permanent binding of centanamycin. Centanamycin, which is our secret sauce, our compound, is cytotoxic, so FDA is going to want us to assure them that the bound centanamycin stays bound. This will get us to a multi-pronged inflection point where we will have de-risked both the veterinary and human platform. We will have data on which to base an FDA Interact meeting, and we will be ready to complete the USDA mandated studies for the veterinary product. ImmuNovX has done quite a bit in our short history. We have completed 2 NSF core programs and 3 accelerators. But we know that there's a lot more that we have to get done, and here's the team that's making all this happen. Together, our team has over 135 years of experience. Alexandra Langendorfer is a 30-plus year veteran of the pharmaceutical industry. She has worked in both vaccines and infectious disease. Alex ran Merck's billion-dollar hospital business for a number of years. Onesmo Mapanju is a 25-plus year veteran of the pharmaceutical industry. He was trained in both virology and microbiology. And Onesmo actually worked at FDA in the Center for Biologics and Evaluation and Research, which is the center that our human product will go through. Sadie Gillespie is a seasoned professional. She has worked in many roles, including leadership roles at Lenovo. Aruna Rajan is a seasoned market research analyst, and she has over 20 years of experience, including at Illumina. I am a serial entrepreneur. My career spans 30 years, and I spent most of my career at Merck, but I'm now full-time on ImmuNovX. You see our esteemed scientific advisors on the right-hand side of the screen. These advisors are all steeped in vaccines, vaccinology, immunology, product development, and commercialization. Together, our advisors have over 240 years of experience, all coming to us. We also have the legal, on the legal side, we have Troutman Pepper. This team is building toward our vision of a future world where vaccine-preventable diseases are prevented. So let me close where we began. So ImmuNovX is an antigen manufacturing company that's poised to make better antigens. Our process takes only 2 hours versus weeks or months, while lowering the cost by 25%, and doing so in one step versus many. Our antigens are safe and efficacious, and vaccines that use our antigens will require fewer cold chain constraints. This is a major deal, as I mentioned, and a game changer for addressing vaccine-preventable diseases worldwide, including in the global south. So I ask that you consider partnering with us. There's a lot of good stuff going on here at ImmuNovX, and we'd love to have you on board. Thank you, if I can get to the slide. Oh, there it goes. Questions? Are you still awake after that? Thank you. The question was delivery systems and how we are planning to deliver our vaccine. The answer is that we are exploring various options. Of course, the injectable, but there are other really good options for delivering vaccines, like nasal and sublingual, for instance. Yes?
In terms of cost, your treatment is purified
Oh, go ahead. Did you want to finish? Were you finished your question?
Yeah, and I just want you, you're pitching this as a very rapid, low-cost
Yeah.
treatment, but in aggregate, producing an antigen requires a lot more money.
It does. It does. And so thank you for the question. The question was about our ability to produce antigens, our methodology for doing that and producing them in a way that actually saves money and is, as I've been mentioning, saves money and is quick. And so you heard me say that our antigens, our technology works in 2 hours versus weeks or months. What I'm talking about there is we are playing in, at least for now, the live attenuated space. And how those types of antigens are developed is over time, it can take months to live attenuate them via passaging. Our technology works more like formaldehyde, which you're probably familiar with, which you just douse it with our centanamycin. It basically binds up the DNA, as I described. And the main difference between something like a formaldehyde and a chemical treatment with centanamycin is that the outer formaldehyde destroys everything about. Our technology destroys the DNA, so it's non-replicable, so it's unable to replicate itself, which makes it safer for instance, immunocompromised folks. But the virus or the pathogen, like coccidioidomycosis, is a fungus. It leaves the outer coat intact, and that's the immunogenic part of a pathogen. So that's how it works. Does that answer your question?
So I guess I can sort of restate it. My assumption then is you must be partnering with people who are
Yes.
delivering the live viruses too.
Yes, so we are partnering. Thank you for asking that question. So a little bit about our partners. We don't have a signed agreement yet, but I can't divulge the name, but we are working with a group that is expert in coccidioidomycosis. They are very tied into everything that's going on there, and we will be tied into as soon as we get that contract signed. Does that answer your question? Thank you. Yes.
I have a question. Does your treatment affect the adjuvanticity of DNA in your preparation? And if so, do you have to add other adjuvants
So,
in order to really increase the strong immune response?
thank you. I think your question, and please correct me if this wasn't everything, I think your question is about adjuvants and whether we're going to add an adjuvant or not?
Yeah.
Yes, okay.
Other DNA, you modify the DNA.
Good question.
Does it have adjuvants?
Excellent question. So the promise of our technology is that it won't need an adjuvant, which is why we can say that we think there will be fewer cold chain requirements for a vaccine made from our antigens. Does that answer your question? You're very welcome. Other questions? No? Otherwise, I'll put my glasses on so I can really see you guys. Okay. Thank you very much. Thank you for your attention.